Okay. Thanks, the COFES organizers, for inviting me. My name is Stephen Larson, and today I'm going to talk to you about digital biology and a project that we're working on called OpenWorm. So today we have awesome CAD software that lets us build complex engineering systems like this Boeing 777. It's composed of millions of parts. They're put together by thousands of designers. And we're able to assign to those parts physical properties that let us do simulations and ask what if questions to see the relationships between those parts as they work dynamically together. So this general process we can do for any kind of a complex system. We might do it, say, for the gears of a watch telling time. And when we build a system like this in the computer, we find often that the subparts, once we decompose them, are actually not executing something that complicated. It's actually kind of simple if you understand some basic principles about how gears come together. So this kind of approach is being used to model tornadoes, like shown here, even being used to model things as large as galaxies colliding into each other. Don't worry, this won't happen for billions of years. But the basic principle is that we, like understanding the rules of chess, we have to decompose a problem down into its pieces, understand the moves that they make, and understand the rules that govern the game that's being played out. Well, in biology, this is not so different. So in biology, there are molecular machines which have individual pieces that follow rules, except in this case, the rules are the rules of physics and chemistry that dictate how they work and how they interoperate. So unfortunately, these are only 3D visualizations. You can't have CAD software today that does this kind of a thing. So we've decided to engage in a project to change that. It's called OpenWorm. And we've decided to take one entire organism, shown here, the C. elegans nematode roundworm, it's about a millimeter long, and turn it into a digital model. So we're going cell by cell. The entire organism has only 1,000 cells in its body, and we are decomposing how its neurons work, how its muscles work, and we are putting them together into a model first that crawls and later does everything that a worm can do. Now, a biological system is a really great example of a multi-scale model. There are processes inside processes and systems inside systems. In just one model of one cell, there's at least 16 sub-processes in the particular model shown here. And if you then put those cells together, you get these crazy hairballs like shown here, which is the network map of all of the neurons inside the C. elegans. But if you want to make software to do this, you kind of need modules. You need to break that apart, and you need to have a process for doing that. So we essentially recapitulated the scientific method, where first you start with observations of biology. If you want to understand the electrical charge across the membrane, first you need to understand what the membrane's made of. You need to understand some things about how ion channels work and how charge passes in a cell membrane. So eventually these observations get turned into a basic understanding of the physics that underlie it and get turned into math. And that math gets turned into equations, and they often get published in a paper, and people read about them. A little bit later on, enterprising mathematicians and computational scientists take those equations and turn them into scripts and run them on computers so we can better understand how that system works. But to build something like a whole organism, we need to go a bit further, and we need to turn each of these understandings of biology into code modules that can interoperate with each other. And so in the OpenWorm project, we did that in a sub-project that's called Geppetto, which essentially acts as a board for these puzzle pieces to come together so that we can have in one system of software, on the one hand, modules that would be responsible for DNA replication, like that molecular machine that you saw earlier, and others that would be responsible for showing you the difference of charge across a membrane. So this is what it looks like. This is Geppetto running just one little process for one cell, showing the opening and closing of ion channels and the distribution of charge across a membrane. This is done on the web so that our network of researchers can see how the model works. And this is really important because this is just one process in one cell. Where we have to go is we have to hook this together with other cells that other experts may be able to weigh in on. And beyond that, we need to be able to put that in an anatomical context so you can see that cell relative to the other parts of the worm. And as we go further and further out from this, there's an entire network of folks that we feel need to be able to use this. So what we've done is we've built an abstraction that energizes a whole community to be able to work on this problem. And we think that this is applicable not just to one little tiny worm, but maybe down the road, it's something that is applicable to being able to do digital biology in a more general way that may even be able to apply to help us understand diseases that afflict human beings, so that eventually we can get the same experience of flying through a 777 that we do potentially flying through digital tissue that we understand much better. So we've assembled a network of folks composed of individuals and organizations. If you'd like to find out more and join us, please visit us at openworm.org. Thank you.